Hello, welcome to this lecture on scientific method. This lecture is aimed at students who are starting to do research in science. So let's start with this question, what is science? We know the kind of dictionary meaning that is kind of a systematized knowledge. And when we say we are doing science, we are doing that activity of acquiring that knowledge in a methodical manner, right? But the important point there is what is underlined, that is the methodical manner. So there is a method. What is this scientific method? We know that, of course, when we think about doing science, the first thing comes to one's mind is kind of curiosity. So curiosity is the prerequisite to do science. You have to be curious and you start asking questions. But it is worth pondering that can you ask all kinds of questions? So that's what the point I wanted to make here. If I ask all sorts of questions, would that all lead to science or not? So let us take some kind of a crazy questions. Let's say we want to know what happens after death. Or I want to know what happened before Big Bang. Or let us say I want to know how biology would work in a different universe if there was one, right? So all of these are questions, but you can all see that these questions cannot be answered experimentally. So the important thing is when we talk about science, immediately ask a question, can you think of doing an experiment to find answers to these questions? If yes, then that would lead to science. Only those questions that are experimentally testable are meaningful. Others are questions, but they are not useful as of today for science. So this is an important point. Questions, but experimentally testable questions. So that is the curiosity is the first point. The second point is knowing tractable questions, knowing questions that can be solved or found answers to. And that's the most important to do science. But when we get education in school or college, we are not taught to make questions. What are we taught? We are taught to answer questions. We are given a problem set, find answers. Somebody would make questions for you. Your job is given a question, how to find the answer. We are never taught to make questions, but when we start doing research, that wouldn't work. We have to make our own questions and answer it. Nobody will give us a question. Make your own question and answer it, but that question has to be kind of interesting for the people around. Only then kind of it will be appreciated. So it's important to train yourself to make questions that are tractable or that can be solved, that can be answered experimentally. And it doesn't matter whether it is academic research or you are going to a startup, kind of non-trivial tractable questions are crucial. Only then, if you have non-trivial questions that can be solved, you will have a business or you will have a enterprise, right? Or you could have a research project and you would find you can be a scientist where you would answer questions which are tractable and they are kind of non-trivial. So that are the 2 points, curiosity and having questions that can be answered. And once you have this question, then the third point is you predict the answer. So you have a question which can be experimentally addressed, but before going ahead and doing experiments, you should try to predict the answer. You should ask the question what the answer can be. Given the current knowledge, what the answer can be. So here is where you use theoretical tools or modeling. So given the current understanding, what can we predict? Given the laws of nature, given the laws of the subject that you are studying, and given the current data, available data, all the previous experiment, can we predict what will happen? So this is the job of people doing theoretical studies or computational modeling. And we need, sometimes it might be easy to guess some kind of what might happen, but for all the non-trivial questions, one would need detailed calculations, detailed simulations to actually predict the answer, right? So you predict using theoretical tools, and that prediction is the hypothesis. In other words, you have a question and you have to frame a hypothesis, and that is done using theoretical tools. So using these tools, predict the answer and frame a testable hypothesis. Here I want to make the point about experimentally testable hypothesis. There can be different kinds of hypotheses. Hypotheses can be right or wrong in quotes, which is fine, which is a part of science. So science goes ahead by figuring out whether a hypothesis is likely to be correct or likely to be wrong, right? So it's okay to have a wrong hypothesis, but what is worse than wrong is to have a hypothesis that is not even experimentally testable. So that's what often called as not even wrong. So it's an interesting phrase attributed to Pauly. So you should go and read about this. You don't have a hypothesis which is falsifiable. So this word falsifiable is the crucial thing in science. You have to have a hypothesis which can be either proved to be right or proved to be wrong. So this has to be falsifiable. Otherwise, it has no meaning and it wouldn't be science. For something to be science, this is the condition. The hypothesis must be falsifiable. Either by you will falsify, or somebody will falsify, or you would prove it right, or somebody would prove it right. But you must have a hypothesis which is falsifiable. Once you have this falsifiable hypothesis, you test the hypothesis by doing experiments. Now, when we test, it may not be just one hypothesis. There will be alternate hypotheses, and we need to test those hypotheses too. And we know that then we would prove or disprove or accept or reject the hypothesis. I put prove or disprove or accept or reject in quotes intentionally, which will be clear in the coming slides. So if the hypothesis is wrong, then you can make a new hypothesis or a new theory. If it is right, then you have to also think about alternate hypothesis. If your hypothesis is right, and if there is an alternate hypothesis that is also right, then you have to determine which one is correct. So that's more work. So the science, as a part of scientific process, we have to figure out which of these hypotheses is correct. And therefore, it's important to think and address alternate hypotheses too. So even if you find a positive experimental result, will you conclude that your idea is scientific? So it need not be. And as we said, the alternate hypotheses need to be tested and checked. When we talk about alternate hypothesis, something that came to my mind immediately is a news that came in press a couple of days ago, maybe sometime this week. So it is based on a research paper which says that they found phosphine gas in Venus. So phosphine is typically a gas that comes from bioprocesses. So would that imply that there is life in Venus? Can we conclude that there is life in Venus? So to conclude, actually we have to test all the alternate hypotheses, and we have to see if any alternate hypothesis would produce this gas in Venus or not, and only then we can say something about life, right? So this is an interesting test example which you might want to read to think about and understand what is the importance of alternate hypothesis to make conclusion. This article does talk about some alternate hypothesis. Do read and do think about how this is important to make a conclusion. Now, you could do an experiment and you might find something, but it could be wrong. So there is an interesting old example. So this is an example from 17th century. So at that time, it's like 1690s or around that time, they didn't know how heredity worked, how inheritance worked. So they had a hypothesis that the male reproductive cells contain a mini human, a preformed human, which is fully formed but it's in a miniaturized form. This was quite popular at that time. And this was called the preformation hypothesis. So they actually wanted to test this hypothesis. So when it is around that time that they invented microscope. So they looked sperm cells and reproductive cells under a microscope. And under a microscope, they actually saw, or they claimed that they saw, a human face inside the sperm, it seems. So that's the claim, at least, right? So what you see on the right hand side is a picture drawn by Nicholas Hartsocker, the person who actually invented microscope. And when they looked at, they actually saw, they claimed that they saw human face, something like a human figure inside the sperm. That's the claim, right? So would you consider it experimentally proven? Of course not, right? So this is nicely described in this book, The Gene by Siddharth Mukherjee. So I want to quickly read out a part from the book. So let me read. The theory was so seductive, the theory of preformation, so artfully vivid that even the invention of microscope was unable to deal the expected fatal blow to the homunculus. In 1694, Nicholas Hartsocker, the Dutch physicist and microscopist, conjured a picture of such a mini being. Its enlarged head twisted in fetal position and curled into the head of a sperm. In 1699, another Dutch microscopist claimed to have found homuncular creatures floating abundantly in human sperm. As with any anthropomorphic fantasy, like finding human faces in moon, the theory was only magnified by the lens of imagination. Pictures of homunculi proliferated in the 17th century, with the sperm's tail reconceived into a filament of human hair, or its cellular head visualized as a tiny human skull. By the end of the 17th century, preformation was considered the most logical and consistent explanation for human and animal heredity. Of course, we now know today that's not right. The experiments were wrong, and they found something which is not there, or they claim to have found something which is not there. So this is an important point to remember, that claims of seeing something experimentally may not settle your hypothesis. It has to be reproducible. It has to stand the test of time, and you have to examine a bunch of alternate hypotheses, and you have to reject all of them. And only it has to be consistent with everything is known, right? Only then this will be meaningful. You may have also heard of some kind of evolutionary argument. This was selected by evolution. So I'm going to tell a joke. So it's a joke. Don't take it seriously. So joke is something like this. So the question is, why are apples falling down? Why are the apples not going up? So the kind of evolutionary reason for this can be argued in this joke is the following. You say that long time ago, there were apple trees that produced apples that went to all kinds of places, right? Some of them went down, some of them went up, and so on and so forth. But those apples that went up could not actually fall to earth and grow again. Therefore, they were not selected evolutionarily. Of course, this is a joke, but think about this in the context of scientific method. So that, again, I want to stress the importance of alternate hypothesis, the importance of testable predictions. And the bottom line is, any argument that you make has to be testable experimentally, has to be falsifiable. So the falsifiability is the keystone of scientific method. Now, if you have a hypothesis and you found one experiment that kind of agreed with you, would that be kind of taken as evidence? So now, answer is of course not. So this is the point where you should understand the probabilistic nature of things and statistical view of things. So you must get trained yourself in statistics even for doing experiments. So please do take a statistics course. If you are a biologist, do take a biostatistics course and train yourself to understand your data and analyze it such a way that you can make kind of statistically relevant meaning from this. So, so the aim of this slide is to stress the importance of probabilistic view. Any hypothesis, we can only talk about the probability that the hypothesis is right or wrong. You don't want to, you shouldn't talk about the hypothesis itself is right or wrong. You should talk about how likely that it is right or wrong. That is the correct way of doing, right? So let us take an example. Let us think about an example which is kind of relevant for the current times. Imagine that you did a COVID-19 test, let's say an antibody test, and you found positive. This kind of would imply that you had COVID. So by just doing one test, will you conclude that you had COVID? Or if you kind of conclude it, will your conclusion depend on whether you are in Mumbai or in Goa or in New York or in Japan? So naively speaking, you did an experiment, that is the COVID-19 test. You got the result. You found positive. Now it shouldn't depend on where you are, but actually it will. So that's where again you want to understand about statistical inference, so that given the data, given the experimental data, you can make the right conclusion. So please learn about statistical inference. Confidence intervals, p-values, these are all part of reaching conclusion from experiments, Bayesian inference. The bottom line is, after all this analysis, the result of a scientific process is essentially a statement about how likely your hypothesis is right or wrong, right? So this is the bottom line of science. At the end of all this prediction, hypothesis, experiments, you will make a statement which would say that the hypothesis was 95, the probability that the hypothesis was right is 95%. Or the probability that the hypothesis is right is 98%. So that is the statement that you can make. Now, you have one likely correct hypothesis. So you have a we have so-called correct hypothesis, but that will have many implications and that would lead to many possibilities. For example, there is a famous Michelson-Morley experiment that proved, or that kind of experimentally shown, that speed of light is a constant, and that had many implications, and Einstein kind of worked out all these implications, and that is what became many of his famous papers, right? So experimental finding or a correct hypothesis will have a lot of implications, and you have to examine all these implications to prove your basic theory right. So therefore, it is important to remember that science is not like a quiz. It is not like a short answer examination or test. It is a time-taking process. It is not a one-day match or a small show. It is a time-taking process. The hypothesis and ideas have to stand the test of time. So as a student, as a scientist, it is important to understand the necessity of a body of work. It's not just like one work. Only a body of work can establish something. One paper may not be enough to prove something. If you have an idea, you might need a body of work to prove all aspects, different aspects emerging from this kind of key idea or key hypothesis. You examine all of this, and only then you will be able to prove some idea or establish an idea. So it's important to think about this idea of a body of work. So we said this curiosity leading to science, answerable or experimentally testable questions, and then you predict the answer using theoretical methods, and that's your hypothesis. So you frame your hypothesis, you experimentally test your hypothesis, then, as we say, accept or reject the hypothesis. Rather, more accurately, you would make a statement that how probable that your hypothesis is true. This is what we aim to achieve at the end of this process. Then you look at all the alternate hypotheses and their implications, and you make a body of work, and then you would have kind of repeat the cycle, right? So this is the kind of scientific method, the methodology of science or acquiring knowledge that we are familiar with and that a research student must know. It is true that scientists need to establish a systematic, methodical, stepwise process, but when we talk about discovery or innovation, many a times it may not happen in a methodical manner. It may not happen in a linear fashion. Often what happens is you discover something by chance. You must be doing some experiments, hoping some hypothesis. You would find completely something different. So you would find something by chance, and later you would connect the dots. But it's important to connect the dots and make all the steps and understand what you found by chance and connect it to what is known. But it is a fact that many findings are found by chance. Hence, one must keep in mind, or one should understand, the importance of curiosity-driven exploratory science as opposed to kind of hypothesis-driven science, where you always have a hypothesis. You may not have a hypothesis sometimes. You might be curious about something and you would want to explore. So I just wanted to say a few things about this. But before that, I recommend this very interesting talk by Yuri Alon. And you could just Google search in YouTube that about this talk. The talk title is "Why Truly Innovative Science Demands a Leap into the Unknown." So search this in YouTube. It's a TED talk. It's a very interesting talk, and I recommend that you all watch this. So when we do this curiosity-driven science, or when we do kind of exploratory science, we'll have tractable questions, and you would explore by doing careful experiments, and you will see what you get. And once you get some data from careful experiments, you may not have a hypothesis at that time, but you did some experiments, you got some data, you wanted to see what will happen if I do this experiment. And you got something, then what we do is that then we have to do theoretical modeling and test various models and check which model would fit this data. If you were to do exploratory research, then the step would be to do experiments, then build models and find the model that fits the data. So you do theoretical computational modeling after doing experiments, but then you ask the question, what is the model that fits the data? Given the current knowledge, given the information that we have, given the laws of nature, what is the best model that fits this data? That is the question that you would ask at that point of time. And it will have implications, and you will have predictions. So then you frame hypothesis. Again, you can frame new hypothesis, and the cycle will continue. When we talk about innovation, this is basically finding something non-trivial. There is, apart from all this method, there is a kind of an element of creativity and imagination involved. So people typically talk about art as creativity, but it is important to think, or in my view, there is also some kind of creativity and imagination involved in scientific discovery as well. And think about some context that I'm saying. So you might have heard story of Einstein versus Hilbert, Darwin versus Wallace, Mendel versus Nageli. So read about them, see how imagination, creativity, luck play some role in kind of scientific discovery. If, for example, in the case of Einstein and Hilbert, if expertise or kind of theoretical knowledge or mathematical knowledge is alone what mattered, Hilbert would have discovered the theory of relativity. So similarly for Mendel versus Nageli, Nageli was a academic, he was a professor, he was trained to do all this. Mendel wasn't, but Nageli couldn't even appreciate this. So the imagination and the creativity does matter. So even in scientific discovery, there is this creativity and imagination. I want you to think about it. Often it's not discussed when we talk about scientific method somehow. So to summarize, you have curiosity and you have set of answerable questions, and you make predictions using the theoretical tools, and that will be your hypothesis. You test your hypothesis using experiments, and then you say that how probable that your hypothesis is right or wrong, and you will have alternate hypotheses and implications, and this will continue, right? But do explore and do leap into the unknown, and that will help everyone. Thank you for listening.